Good evening, good morning, good afternoon, everybody. It is actually 7 p.m. in Paris, 6 p.m. in London. It's noon in Houston. It's 8 p.m. in Patras. And it is my great pleasure and honor to welcome you all to this journal club by the International Academy for Clinical Hematology. This is our first journal club after the summer break, as you know. And of course, we are very grateful to all of you attending today this first journal club after the vacation period. As you know, these journal clubs have an unrestricted educational grant. The steering committee has chosen a very important article, in my opinion, for today's journal club, and this is a JCO paper just published a few weeks ago, and this is about the long-term benefits of Tagraxofusp for patients with blastic plasmacytoid dendritic cell neoplasm. And I believe this paper is really important for several reasons. One important reason is, of course, about this ultra rare orphan disease, unmet medical need, but most importantly, and I think the authors should be commended and praised for this, is that we have long-term follow-up. And I think more and more, this should be the rule. So beside the initial results published in a lovely journal, we need to have this culture of, publishing long-term follow-up. So this is the abstract of this paper. And actually, the idea was simply to give the long-term follow-up about those patients who were initially published in the seminal New England Journal of Medicine paper. And the story is quite straightforward, because in this long-term follow-up, you had 65 patients treated first line, versus 19 patients treated in the relapse refractory setting. And I'd like to emphasize how difficult is this disease, because we're talking about a disease of elderly and frail patient, median age 72 up to 87 being treated here. You can see the skin localization, which is a key feature of the disease, the bone marrow infiltration, lymph node infiltration, but other organ involvement. And interestingly, the relapse refractory patient have received and failed several treatments. And you can see here the response rate. So these are the updated response rate based on the stage of the disease. And you can appreciate that we are talking about CR/CRc of 57%, and overall response rate was long-term follow-up 75%, and 1/3 of the patient were able to be bridged to transplant, to allogeneic stem cell transplantation. And we know very well that this is the only curative treatment in this very hard to treat disease. You can see the median duration of CR, the median duration of follow-up, the median overall survival, roughly 16 months up to 25.8 months. These are really very important results. So I'll stop here, and to discuss these excellent data, we have the pleasure to have Professor Naveen Pemaraju, who has been really the leader who pioneered all of this work and have allowed to raise awareness about BPDCN. So thank you, Naveen, for all the hard work since many, many years. But also we have the other panelist, who is another expert, Professor Alexandros Spiridonidis, hopefully I'm pronouncing it correctly, from the University of Patras, who is also an expert in the management of these BPDCN patients, but also, of course, in stem cell transplantation. So, Naveen, I thought this is really an excellent idea that should become maybe more and more mandatory, you know, having long-term follow-up results for all the trials. So before going into the trials, because this is relatively, you know, I would say confidential rare disease, can you remind our audience about what is this drug, you know, Tagraxofusp first?
Yeah, well, thank you, Professor Moti, for your very beautiful overview, very succinctly kind of addressing some complex issues. I really love the way you teach, and I want you to know that that was wonderful. I think where we start today is, as you said, BPDCN is a ultra rare disease. We think maybe only 500 to 1,000 patients a year in America, maybe about 500 to 1,000 across the EU. As you mentioned, the Tagraxofusp or TAG agent that we're talking about here today is the first, and as of this moment, only US FDA approved and EMA approved drug. It's a diphtheria toxin-based drug, which is modified and conjugated to recombinant human IL-3. And so the goal is for this drug to hit the IL-3 receptor alpha, also known as CD123. One other principle is that all BPDCN patients will not only express, but likely overexpress this CD123 target. And so we think it's sort of rationalized molecular targeting in this disease. And then one final aspect, as you nicely mentioned, anytime you have a new drug and you have a new field, as we have, we're proud that we've raised awareness, but we also want to raise awareness that it can have toxicities. And sometimes patients can get into a capillary leak syndrome in the first cycle. And so that's something we know about, we can educate, mitigate, and prevent. So I think that's where we'll start the discussion. A new drug, diphtheria toxin conjugated to human IL-3, and has very nice response rates in BPDCN.
Thank you very much, Naveen. Very clear, very straightforward, and thank you for the kind words. And by the way, guys, if of course we would appreciate and would love to have your questions and comments, and I'll share them with our panelists. And also, please don't forget that we have our annual IACH meeting at the end of the week, and Professor Pimaraju will be delivering a very nice lecture about this rare entity. So, Alexandros, now I'd like to go to you. So you didn't participate to the study, and that's, you know, the principle of the Journal Club, to have someone from outside. So when you saw these results and you know, this paper, what were your thoughts? I mean, you were impressed or you were disappointed with long-term follow-up? What is your appreciation about these results? And obviously, I'd like maybe if you can comment on this issue of bridging to transplant and its importance.
So thank you very much. I must say I was impressed. We could comment later for some, not limitations, but some thoughts that I have. But I was impressed mainly because I knew the other study that was published before in the New Journal of Medicine, which was a study which was, in my opinion, showed a biological activity, but it was not so impressive or didn't show me so powerful that there is an efficacy of this drug. I was impressed now, first of the follow-up, which is mature. Second, of the number of the patients. I was really impressed to see so many patients included, and especially, if I'm correct, there are 40 more patients after the other study finished. So within 1 year, if I'm correct, we included 40 patients, knowing the rarity of the disease. I think in my life I saw 5 or 6 such patients, maybe, well, okay, less than 10. So having 40 more patients for me was very impressive. And so I think that in this study, what impressed me that having so many patients, especially in the first-line treatment, which I mean the first-line treatment for me are more, let's say, definitive results, which I can comment, let's say, convince me that is the way to go for these patients. Of course, there are limitations, we can discuss afterwards, but I show the patients are old, are the patients that we are expecting to see. And at the end, what I it remains to me is that these patients can be cured. Of course, only with transplant. I suppose only with transplant. You have some 2 patients without transplant, but I think it's transplant. And what remains to me is this is a goal. Do something, bring them, bridge them, and bring them to transplant. And as may discuss later, as I understand from the data here, is bring them to transplant as soon as possible. Don't wait because they may relapse very quick. This is data which I try to understand from this study. So at the end, many patients, efficacy data on first-line treatment, bridging to transplant, transplant is a curative option. And in comparison to the old study, this is not a stage 1, 2, 3, 4 cohort study, where in the old study, I think the point was to show the toxicity or the dose and other issues. This was as mainly in point, bridging transplant and efficacy. So
excellent.
Nice
before going into the limitations, Naveen, can you comment on the results? Because it looks to me that, first of all, the long-term results are really consistent and they're even improving over time. But also, and I think this is crucial when it comes to sort of a long-term follow-up, is that there are no safety issues apparently, because initially there were some concerns about the so-called capillary leak syndrome and maybe other toxicities, but it looks like now this is not really a big matter of concern. Can you comment on the results?
Yeah, I really appreciated both of your analysis, Professor Alexandros. Very nice comments. Agree all. As you both mentioned, the strength is this long-term follow-up, which we were delighted to see, approximately 3 years. And as you mentioned, this overall response rate holding steady, 75% with a 57% CR/CRc. With this type of follow-up, I agree with both of you. This is impressive. The 2 aspects that Professor Modi just brought up are key. One is I was looking to see if there were any new safety signals, which I'm proud to tell you there weren't. So sometimes we know in these novel drugs you can uncover. And then second, what about this capillary leak syndrome, which we did see was fatal in the original experience? And that's fantastic. So as you'll see, the rate stayed the same. It's approximately 21% is the rate of capillary leak syndrome in the frontline patients. It's exactly what we expected. Almost never is it fatal anymore now that we know watch the albumin, replace the albumin, watch the fluid status and the weight. And then the other 2 toxicities that we see, usually only restricted to cycle 1, there's a transient thrombocytopenia in about half the patients and a transient transaminitis in about half. Usually don't see that in the subsequent cycles. And importantly, not a high rate of neutropenia or neutropenic fevers. That was one of the worries with targeting CD123. So no hair fallout, no real nausea, vomiting. Older patients are in pretty good shape. And then as Professor Alexandros nicely mentioned, get them to stem cell transplant as soon as possible, even if the age is 70 or 71. That's an impressive part of the long-term follow-up here.
Excellent. Thank you very much. So, Alexandros, so we mentioned, and I was very pleased to hear the word cure, because usually these BPDCN patients are usually old and frail. So they are not the usual or optimal candidate for an allo transplant. But here, I think, especially among the CR patient, we have 50%, I think 51% who were able to be bridged to transplant. So maybe, I don't know, Alexandros, if you can comment on this and whether this 51% rate, in my opinion, it's quite high, because usually it's much lower. What I know from the literature and from registries. What are your thoughts here?
Well, I was impressed to see patients that first they went to transplant, and also they were all patients. I saw also the population that were included. The most of them, as we expected, were all patients. So, of course, in a non-randomized trial, there is selection bias, but this comes not very clearly out that there's a selection bias. Maybe you can tell us also what was the patients that were transplanted. Probably Of course, there was a selection of them, but I was amazed to see that so many patients were transplanted, and also long-term results after transplantation that they are cured. Because probably we don't know this disease very well, but we expect also for AML patients that they relapse. I would expect more relapses after transplantations, and we don't see this, even for the patients. My question here is probably the patients that went to transplant were all patients that were in remission, I suppose. Although, if I read it correctly from some supplementary material, there were also some patients that they were not in remission because they achieved the CR after transplant.
Right.
So my question will be here, transplant, okay, is curable, a few relapses after transplant, but I was wondering, can you find out which patients do really need not this transplantation, which go well with the transplantation? If there are some patients that we do know that they don't go well, and we can assume that there are patients that we expect that they go very well. I'm not sure that we can find with this data, this patient, but this was the question that came out to me. Can I somehow find out which patient should I go to transplant?
And then maybe Naveen, I would like to add here a question we have received from the audience. Obviously, we can't take all questions, guys, because we are quite limited in time, and you can see Professor Pumaraju is in the middle of his clinic. So we have a question from, I think, Dr. Suarez Uriel, who is very loyal to the Journal Club, about how do you assess a response, because we know, you have this skin localization, sometimes you have the bone marrow involvement. So let's try to have a response to this issue of assessing the response in an objective way, and what level of response you would require to bridge them to transplant.
Well, that's great. So yeah, I think that both questions kind of go well. So from Professor Alexandros's observations, all correct, 100% what both of you said. I personally, I'm looking at this data every day, I'm amazed. We took patients who are older and frail, and some of them, the performance status improved, gentlemen, over time. So I give the Tagraxofusp agent, maybe you're a PS of 1, now you're more of a 0. It's incredible. So you're right. So older, frail patients actually became even more stable. Most everyone was in a nice response, but as you said, there were 2, 3, a few patients who were not in a response who still benefited post-transplant. So as you said nicely, selection bias, it is a clinical trial. So you already have to get on the clinical trial, right? You have to be healthy enough. And then we have a nice transplant team with my friend and colleague, Dr. Muzaffar Kazelbash, ready to transplant. But the majority of these patients are allo transplants in older patients. So for the response criteria, it kind of ties into the transplant. We had to invent them. In this ultra rare disease, when we first put this together 10 years ago, there was no response criteria. So what we did, and it's reproducible. I use it in clinic. We use it in the subsequent clinical trials. I think there's 3 major compartments. One is for the skin, of course. We borrowed from cutaneous T-cell lymphoma, mycosis fungoides. So that's the MSWAT criteria. That has worked beautifully well. So now I really work hand in hand with the dermatologist. So we understand that. So you make the subjective objective, and then you can replicate it over time. 2, we use lymphoma staging for the lymph nodes, so use imaging, either PET scan or CT scan. And then 3, bone marrow, and using our standard AML criteria. So put all that together. A CR has to be complete resolution of all compartments that were involved at baseline, so whether that's 1, 2, or 3. And then we did denote a CRC, which is a bit different from AML and other things, which just reminds you that you can have complete remission, but you do a biopsy or you have 1 residual skin lesion left, it's scarring, it's not disease, it's something left over. Those people do very well. So I think that's the response criteria. And finally, yes, you can transplant patients, even if they're not in a CR in every single compartment, as long as you think you have a debulking of the disease, and then go forward. So I think those are kind of the unique aspects of BPDCN.
Excellent. Thank you very much. We have a question here that I will direct maybe to both of you, because this long-term follow-up study included patients who were in relapse refractory setting. And I must confess, I was personally very surprised to see, for instance, that you had 2 patients who received 3 prior lines of therapies.
That's right.
2 patients who received more than 4 lines of therapies. You have 3 patients who receive 2 lines of therapies. And usually my understanding is that we have very limited options in these patients. Either you do a sort of an ALL-like regimen if they can receive an intensive chemo, or an AML-like, and then unfortunately either you fail or you achieve something. So today, Alexandros, in your practice, I know it can't be routine because it's very rare. What are the options for the relapse refractory patient? Because I believe these results are quite appealing, you know.
Today, we used to use AML type therapy, but we were thinking about the venetoclax type therapy nowadays. Also, I think there's also a clinical study on that. And there are some reports that these patients may, let's say, respond to venetoclax. So because of also old patients, if I have one relapse refractory patient, I would think if I don't have the possibility to give this drug, I would also venetoclax-based therapy also, in my opinion.
Excellent. So Naveen, we have a question about the modalities of this treatment in the relapse refractory. So we have a question, for instance, can it be given, for instance, after transplant? Because some of these patients may relapse after transplant, unfortunately. First of all, that's one question we have. And the other question we have is about what is the safety profile in those patients who received and failed, for instance, 1 or 2 lines of therapy. Nice. Obviously, today, my gut feeling is that we should use it frontline, but, you know, as Alexandros mentioned, it may not be available everywhere or affordable, and this is why we have to live with the reality. So how did it work for these relapse refractory patients?
Yeah, you guys, this is a great discussion. So I think this is going to be helpful to folks out there. Everything you said is right on the money. So for the relapse refractory post-transplant, very dire situation, whether it's AML or in particular this BPDCN, as you said nicely, Alexandros, yes, venetoclax-based therapies. So either older AML paradigm, comparing it with HMA venetoclax if you can, ALL-based regimens, as Professor Modi and the French have pioneered very nicely. So hyper-CVAD in the younger, older, lighter chemotherapy. And then there's also 2nd generation CD123 agents. So we should mention that there's another program called IMGN632. So that's pivekimab sunirine in US and Europe in clinical trials. And then there's CAR-Ts and all these things. But specifically post-transplant, yeah, you're worried about safety here. GVHD, LFTs, VOD, you're really watching these patients. We have given the Tagraxofusp agent successfully and feasibly, either as a maintenance program, a post-transplant, or in frank relapse. So safety concerns are the same, watching out for LFTs, platelets, capillary leak, but the stakes are a bit higher because you have post-transplant to also think about. So we have given it safely. You can do it. I think that's a great move. And then second, as Mohammed is nicely bringing up, sometimes I'm not able to give the CD123 frontline. And yes, it is okay to give it relapse refractory. Obviously, you'll see from the New England Journal and JCO, the response rates are not as high as we're used to in these myeloid diseases. But you can still get some efficacy. You can still get a bridge to a transplant or a second transplant. Again, safety is a concern here. Maybe a little bit higher chance for capillary leak because the patient may not be as fit. You're really watching the albumin. You need to be 3.2 to get on the drug without albumin, but it is okay to supplement with IV albumin once you get on the drug on your own. And then I think the other issue is also overall survival. Frontline, obviously, we're pleased with the results compared to historical, but in the relapse refractory, we still have work to do. And what both of you are reminding me is it's still about cure. We've got to go for combination therapies and stem cell transplant, optimizing all of these. So I think monotherapy is great in the front line, and it's a great step forward, but cure still needs either the transplant or combination therapy. That's where I think the field is moving.
Okay, excellent. Well, one question which is maybe goes beyond the scope of the paper, but I will take it. For you, Alexandros, when it comes to allotransplant, how is there a preferred conditioning, for instance, a TBI-based conditioning because of the extramedullary localization? And what are your thoughts about this?
This is exactly what I wanted to ask.
But you are the transplanter. I mean, Naveen said you are the transplanter. he wanted to avoid this question.
Okay, so I'm not a TBI fan. So of course, because it's extramedullary disease,
right,
we could think about that, probably lower TBI dose, not the 12 Cray, because they are frail patients. But because they are older patients, I was thinking something about the reduced toxicity, conditioning, what's in every center the favorite is, something like that. We don't know.
Exactly right. And oh, let's see here.
So nothing special as a recommendation, I would say. Whatever would allow the transplant, I would say, the graft to get in, we would take it. But obviously, for instance, I can mention that because of skin localization, you have drugs like thiotepa, the conditioning that they have a good penetration in the skin, for instance. So one last question to you, Naveen, because I know you are in a hurry in your clinic, and of course, our philosophy is always patient first. Despite the lovely educational activities, patient first.
That's right.
So what's the next step now for BPDCN? So you pioneered this wonderful new agent, and it's really bringing hope and helping many patients. What are the next steps? How are we going to move in this rare entity while we know very well that we will not be able to do studies with thousands of patients?
Thank you, Mohammed, for this opportunity. I would divide it into three major categories for next steps. So one is improving on the CD123 targeted field that now we have. So better drugs, safer drugs. And so I have a lot of high hopes for this current trial that we're doing, the IMGN 632. So let's see how those data go. And we've presented those publicly before. The second step is combination therapies. So we're very excited about the triplets that we're now doing in the clinic. So younger patient, hyper CVAD, CD123 agent, venetoclax, older unfit, CD123 azacitidine, so HMA venetoclax. So moving those into the frontline setting, I think, is a very exciting option. And then third, Mohammed, I want to emphasize here, central nervous system involvement. We were surprised to see, and we published on this, a very high rate of CNS in BPDCN. So not like AML, more like Burkitt's or high-risk ALL. So we have incorporated lumbar punctures into every patient, frontline or relapse, and I think that's helping catch and to prevent. I think these three help you to get to a total therapy, triple therapy, like you and others have done in multiple myeloma, for example. That's where I envision our field going, that we move more therapy, smarter and better upfront, prevent MRD, prevent CNS, prevent relapse, and maybe even, Alexandros, put you and your team out of business and eliminate the stem cell transplant and go for cure. We're not there yet, but we're working on that. That's my vision for the next 5 to 10 years.
Fantastic, guys. I think you did a great job. We can spend hours on this. This is fascinating, but as I mentioned, we do have the IACH virtual meeting, free registration to all guys, and there is a special session dedicated to the rare entities, including BPDCN. So you can connect on Thursday and Friday. And I think the timing of these sessions is quite okay to allow you to connect whether you are in North America or in Europe, because of course time zones are always difficult to manage. So I hope you have enjoyed this journal club by the IACH. I do apologize, we were not able to answer all the questions, but I'm sure you can email our fantastic faculty, Professor Pemaraju, Professor Spyridonidis. So wherever you are, please stay safe and keep well, and see you in a couple of days at the IACH annual meeting. Thank you very much. Take care.